The study of the human metabolome now provides novel and unique opportunities to explore the impact of the environment on body function and health, along with the impact of individual genetic variation. The metabolome is defined as the totality of small molecules in the body with a molecular weight up to 1500 Dalton. The metabolites that we find in our body are a combination of endogenous traits from our own metabolism as well as exogenous factors such as diet, gut bacteria, drugs that we are exposed to, food contaminants and additives, as well as toxins. By studying these small molecular profiles, we can attempt to characterize biochemical pathways, exposure to environmental factors, as well as genetic variation. We do this with the goal to better understand the metabolic mechanisms that are underlying health and disease, and we may also be able to determine biomarkers that reflect both exposures and outcomes. If we compare metabolomics to other omics techniques, metabolomics, or the metabolome, is the one omics measure that is closest to our phenotype and to our health and disease outcomes. Our genes obviously determine the genotype. The transcriptome is determined by epigenetic modifications of our genes, as well as transcription factors and other regulators of gene expression. How and to which extent the genes are read and the products of genes, peptides and proteins are produced determine the proteome. Many proteins that arise are enzymes that modulate our metabolism and thereby our metabolome. Therefore, the metabolome is closest to our phenotype and to health and disease outcomes. Our metabolome reflects our biological phenotype and is modulated by a variety of factors such as sex, stress, inflammation, disease, drugs, diet, and microbiome. Each one of us and each specific situation should have a specific pattern of metabolites which depends on our genome, epigenome, transcription activity, lifestyle and physical activity, diet, inflammation and diseases, and not the least, our bacterial or gut microbiome.
Methodological approaches to do metabolomic analysis. One approach is untargeted analysis. The concept there is to try to collect all signals that can be measured and analyze these patterns without the need to identify all of the individual compounds. Specific patterns can then serve as biomarkers. And if you find an interesting feature that is a useful biomarker, for example, for a disease, a second step, you can then try to identify the biochemical nature of these signals. In our group, we have followed an alternative strategy of using targeted metabolomic analysis. Here, we aim at measuring quantitatively known metabolites as precisely as possible. To meet this aim, we use liquid chromatography coupled to triple-quadruple mass spectrometry. That is similar to the methodology which is used in the neonatal screening for inborn errors of metabolism in many countries of the world. Our targeted platform covers phosphatidylcholines, sphingomyelins, lysophospholipids, non-esterified fatty acids, tricarboxylic acid cycle intermediates, amino acids, carbohydrates, and many other compounds. The beauty of the method is that you can measure hundreds of metabolites from a single drop of blood. Typically, we use as little as 50 microliter of plasma. Using a standardized methodology, we can measure a large set of metabolites based on a variety of different methods, including amino acids, phospholipids, and many more. It is also possible to measure those metabolites in any variety of different matrices, such as urine samples have the advantage of being non-invasive. This is of particular benefit when samples of young children have to be analyzed.
we applied our metabolomics platform in a large number of studies of pregnant women from the US, from the UK, from Ireland, from Spain, from the Netherlands, and from Germany. Additionally, we collected some cord blood also in a number of these studies. We did this to analyze how maternal exposures predict the maternal and offspring metabolism and maternal and offspring health. The repeated analysis of all these large studies is necessary in human mechanistic studies to provide evidence. For instance, in collaboration with Patrick Wadwa and Sonja Entringer from the University of California, Irvine, we showed that the metabolome changes during pregnancy. And we also found that pre-pregnancy BMI, so the obesity status before conception, has a strong influence on the metabolism during pregnancy. Even stronger compared to maternal diet in pregnancy. In this slide, you can see in the outer parts of the circles, the lines and points highlight strong association between the predictors and the metabolites, which are arranged in circles. The effect of pre-pregnancy BMI was strongest for non-esterified fatty acids, which are a long-term dietary marker. So we could show that preconceptional exposures affect the maternal metabolome and even offspring metabolome and can be related to the offspring health. In another study of pregnant women affected by gestational diabetes, we could show that there is an impaired transport of biological important polyunsaturated fatty acids. DHA, the most important biological essential fatty acid, is well transported from the mother to the placenta. But the transport from the placenta to the fetus is reduced. The fetus has an impaired DHA status. This is of importance, so we could show that gestational diabetes is really affecting the fetus and the fetal health and metabolism. And it is possible that women suffering from gestational diabetes need more DHA during pregnancy. Since also other polyunsaturated fatty acids are affected by these mechanisms, there are further study needed to explore how all these fatty acids are transported from the mother to the fetus.
Another important question in pregnancy, as discussed before in other videos, is the market benefit of the supply of omega-3 DHA for reducing early preterm births before 34 weeks of gestation. DHA supply has been associated with protection against child allergy and with improvement of the child's visual and neurological development. Our metabolomics techniques allow us to measure very precisely the incorporation of dietary DHA into the lipids that are found in cell membranes of tissues such as the brain. When we gave a large dose of half a gram of DHA per day to healthy people, we could follow how fast this DHA is then incorporated into the cell membranes of red blood cells. This helps us to predict the optimal strategy of DHA supply to support adequate incorporation into the tissue membrane lipids. The metabolomics technology offers us very exciting perspectives for new windows of opportunity to improve our understanding of the physiology and pathophysiology of pregnancy, as well as the impact of diet and lifestyle on maternal and fetal health. If we enhance our insight into the underlying biochemical mechanisms, we should also find even better opportunities for targeted and more effective preventive approaches.